This lesson's on the electron transport chain, and here are the objectives. So to review what we've seen so far in aerobic cellular respiration, in glycolysis we saw that glucose was partially oxidized to pyruvate, making 2 ATP by substrate level phosphorylation, and also making 2 NADH per glucose. And then in the Krebs cycle, pyruvate was completely oxidized to carbon dioxide. At this point we have no more of the carbons that we started off with from glucose. They've all left as carbon dioxide. 2 more ATP were made per glucose by substrate level phosphorylation, but we also made some more high energy electron carriers, 8 NADH and 2 FADH2. So that was glycolysis and the Krebs cycle, and now we're going to look at the electron transport chain, where we're going to now use these high energy electron carriers to make ATP. So the electron transport chain is made up of proteins on the inner mitochondrial membrane. So we're here along the inner mitochondrial membrane. And actually I say proteins, but there is one of the molecules that's not a protein called coenzyme Q. But mostly proteins, and we're on the inner mitochondrial membrane. And what's going to happen is that electrons are going to be passed from these NADHs and FADH2s that were made in glycolysis and in the Krebs cycle. They're going to get passed into this chain to more and more electronegative carriers. Remember that something that is electronegative or highly electronegative means that it's greedy for electrons. It wants to hang on to electrons. So let's say, for example, I had 2 different proteins. I'm going to call these proteins protein 1 and protein 2. And let's say that protein 2 is more electronegative. I'm just going to write EN for short. And this is an electron. If I could pass this electron from protein 1 to protein 2, because protein 2 is more electronegative, this is going to be a more strongly bound state. Protein 2 is hanging on to that electron more strongly because protein 2 is more electronegative. So doing this, passing the electron from a less electronegative to a more electronegative protein, is going to release energy. So that's going to release energy. You can think of it like rolling a ball down a hill. Here, this is state 1, and this is state 2 down here, and you can imagine rolling a ball down a hill. If I roll that ball down the hill, because state 2 is a more stable, lower energy state, then energy has been released here. And in this case, the energy is being released as kinetic energy, the energy of movement. But in this case, the energy could be released in another form, and we'll see that in a moment. So the principle here is that we're going to pass electrons to more and more electronegative carriers, and that's going to be exergonic. It's going to release energy because we're moving the electrons to a more and more stable state. At the end of this, we're going to want to pass the electrons to something very, very electronegative and get them to leave the chain entirely. And for that, we use oxygen. Oxygen is the final electron acceptor in the chain. It takes the electrons off at the end of the chain because it's very electronegative. If you think of your periodic table, it's in the top right-hand corner, and atoms in the top right-hand corner of the periodic table are very electronegative. So by passing these electrons to more and more electronegative proteins, we're going to release energy, and that energy is going to be used to pump hydrogen, to pump protons, into the intermembrane space. The intermembrane space is the space that's between the inner and outer mitochondrial membrane. And so we're pumping protons into this space in here that I'm shading in red. We're going to pack a bunch of protons in there, and doing that requires work. It's endergonic. It takes energy. And that's both because we are creating a concentration gradient with a high concentration of protons in the intermembrane space and less of them in the matrix, and also because there's an electrical repulsion between all the positive protons that we're trying to pack into one space. So by packing all those protons in there, we're actually storing potential energy in the proton gradient. So the energy, which was originally in food, then is in the high energy electrons in the NADH and FADH2, has now been used to pump protons into the intermembrane space and is now stored as potential energy in that gradient. In other words, those protons are going to want to get out of there and get back into the matrix if they can, both because they're repelling each other electrically and because they want to move from an area of high concentration to low concentration. And when they do that, when they move from the intermembrane space into the matrix, that's going to release energy, and we can use that energy to drive energy requiring or endergonic reactions, and specifically to make ATP. So let's take a closer look at this now. Here is a schematic of the mitochondrial inner membrane. You can see that we've got the intermembrane space. This is the part that I've colored in red above, and down here I have the matrix. And in the gray here, these are all the molecules, again, mostly proteins, of the electron transport chain. And they're going to get more electronegative as we go to the right. So the ones on the right are going to be able to strip electrons off the ones that are immediately to their left, again, because we're getting more electronegative as we go to the right. The white area is just the phospholipid bilayer. And over here, this is an enzyme that is not part of this electronegative chain here. It's an enzyme called ATP synthase. And I've tried to show that it could be elsewhere in the mitochondrial inner membrane by drawing a break here. This is not actually a true break in the mitochondrial inner membrane. The membrane is intact, but this is just showing that this could be in another place. So if we start with NADH, and remember this NADH has come from either glycolysis, so it had to get from the cytoplasm into the matrix, or it was made in the matrix in the Krebs cycle. It's going to feed into the first protein here, and it's going to be oxidized to NAD+, and its electrons are going to be passed into the chain. I'm going to draw the electrons here in green, let's say. And so the electrons are passed into the chain, and then they're going to get passed from one of these molecules to the next. And I'll just stop there for a moment. And the NADH lost a proton as well, and that proton is going to be pumped into the intermembrane space. And again, it's the energy release, or the energy that is released from this oxidation. This is an exergonic reaction that's used to pump the protons in here. And again, as they are passed along, more and more energy is being released as those electrons are moving to more and more electronegative molecules. And so there are further spots where protons are pumped from the matrix into the intermembrane space. So there's another one there. Again, the energy for this pumping is coming from the fact that we are moving the electrons to more and more electronegative molecules. Now that's NADH. FADH2 comes in a little bit later into the electron transport chain, but is similarly oxidized and donates electrons as well as getting rid of protons. So the electrons now, here I'll draw them in green, they join the chain as well, and they're going to get passed along. And again, the same kind of proton pumping happens with the energy that's released as those electrons are passed along the chain. Notice that FADH2 enters the chain a bit later than NADH, and so less protons are pumped for every FADH2 than for each NADH. Now, over here, the electrons of both ultimately need to leave the chain. If they didn't, the chain would back up, and we wouldn't be able to pass any more electrons along. And that's now the role of oxygen. So oxygen comes in, and I'm going to show this balanced by writing half an oxygen. Oxygen comes in, and together with two hydrogens, two protons, is going to pick up these electrons. So here's two electrons. I'm going to draw that in green again. And those two electrons together with the two protons and half an oxygen molecule, if you look at that and put that all together, you get H2O, one water molecule. So this is where water is produced in cellular respiration. And you might remember from the equation that we looked at for cellular respiration, water was on the right-hand side. This is where it's being made. So oxygen is called the terminal electron acceptor. The terminal electron acceptor because it is taking electrons off of the end of the electron transport chain to allow it to keep moving. Now we're left with all of these protons that are stored in the intermembrane space. Again, that's a high concentration. Down here there's a low concentration, and also they're repelling each other because they're all positive. So for both of those reasons, they would like to get out of the intermembrane space and back into the matrix. But there are no breaks in the mitochondrial inner membrane, and so they can only go through or get back into the matrix where they're allowed to. And where they're allowed to is through this ATP synthase enzyme, which has a channel in it that allows the protons to fall back down their electrochemical gradient, electro because of the repulsion and chemical because of the concentration gradient. So they fall back down their electrochemical gradient back into the matrix, and in so doing, they release energy. And when they release energy, that energy is used to make ATP. So ADP and phosphate are joined together. That's an energy requiring or endergonic reaction, and that energy comes from the hydrogens moving through ATP synthase down their concentration gradient. And here is where most of the ATP is made in cellular respiration. This is oxidative phosphorylation. Oxidative because we are stripping electrons off of successive molecules. So this is oxidative phosphorylation. One way you can think about the energy coupling that's happening here is thinking about it like you're using electricity to pump water up into a water tower and then letting it fall down the water tower to turn a turbine to do work. That's not what's happening here, obviously, but it's a similar or an analogous situation. So this process is called chemiosmosis. In chemiosmosis, the protons fall down their electrochemical gradient through the enzyme ATP synthase. That's the enzyme that we've shown here above at the right. And that process is exergonic. It releases energy, and that energy is then used to synthesize ATP from ADP and phosphate, which is an endergonic process, or an energy requiring process. So again, we have energy coupling. So I've said that the majority of the ATP that's made in aerobic cellular respiration is made by oxidative phosphorylation at the electron transport chain. And the overall ATP yield for the full process of aerobic cellular respiration is about 30 ATP per glucose. Now you might be wondering, why is it approximate? Well, there's a number of reasons for that. There's not an exact whole number ratio between the electrons moved and the protons pumped and the amount of ATP made. There's a difference between the energy yield of NADH coming from glycolysis versus NADH coming from the Krebs cycle, and the proton gradient might be used to power other work. There's a variety of other factors that make this approximate, and in fact, if you read textbooks, you'll see this number as quoted anywhere from about 28 ATP per glucose to about 38. So we'll think of it as about 30. Whereas the ATP yield from fermentation is only 2 ATP per glucose. Those coming from glycolysis, no further ATP are made in either lactic acid fermentation or in ethanol fermentation, just the 2 ATP per glucose from glycolysis itself. So there's about a 15-fold difference in the amount of energy released from glucose in aerobic cellular respiration versus fermentation. Let's finish up here by looking at our equation that shows the overall inputs and outputs of cellular respiration. And we'll think about where each of these go and where they come from. So glucose was first broken down into pyruvate in glycolysis, and then pyruvate was further oxidized to CO2 in the Krebs cycle. So that's where this CO2 comes from, and that's where the glucose has gone. Now we've seen in the electron transport chain that oxygen is the final electron acceptor. It's highly electronegative, and so it takes the electrons off the end of the electron transport chain to allow the electron transport chain to keep going. So the oxygen is used in the electron transport chain as the final or terminal electron acceptor. And then that oxygen is combined with protons and electrons to make water. So that's also coming from the electron transport chain. We said that the carbon dioxide comes from the Krebs cycle, as well as from the link reaction. Remember that the link reaction makes 1 of those carbon dioxides per pyruvate or 2 per glucose. And the energy is first taken from food, put into NADH and FADH2, those high energy electron carriers, with a little bit put into ATP by substrate level phosphorylation, but then the NADH and FADH2 are oxidized at the electron transport chain, and that's where most of the energy gets into ATP. So that is now a look at the full process of aerobic cellular respiration. We've looked at glycolysis, the Krebs cycle, and the electron transport chain.